even identify it at that level. But in the medical school, the final exam, when I was taking it with as a graduate student, we had to identify 50 slides in 30 minutes as to the organ or peripheral nerve or something. So the more you can do like that, not even bother to take the time to put it in the microscope, the easier it was. So out here is the cortex, and the dotted line is not part of the slide, okay? That's just kind of putting in. So what we look for on a low power view, when we look for cortex versus medulla, is the present, this is probably a 4x or a 10x, and these structures here are all the capsules, with the glomeruli inside and the capsular space showing around it. As soon as you see that capsule, you know that you're in the cortex, okay? Now, a little closer view, you'll see proximal convoluted tubules and distal tubules, and those are only found in the cortex as well. But the capsules are the easiest things to see at the lowest view power. These other structures here are known as medullary rays, and we have renal cortical columns, but you don't have to worry about those concepts, okay? This is what I was talking about as far as the medullary ray. So here is the capsule, so let's look at that a little bit closer. And in here, wherever the black dark dots are, those are red blood cells. And they're going to be covered by the podocytes, but at this level, because it's all twisted and turned, we're not going to differentiate. We do that on an electron microscope, like I showed you during lecture. Then we have the capsular space, and this is the parietal layer of the capsule, so it's also simple squamous epithelium, all right? But it doesn't have fenestra, it doesn't have filtration slits, so we don't worry about particles leaving the capsule and getting out of that. Now the proximal convoluted tubule, remember I told you had very high microvilli, very tall microvilli, and it creates a messy appearance inside the proximal convoluted tubule. So here would be a proximal, over here is a proximal, here's a proximal. Questions? So these would be the long microvilli, and see how clean this is? How the nuclei bulge, even though we do have some tufts. This would be a distal convoluted tubule. Over here is a distal. Okay, here's a distal. So they're distinctive from each other. Now this is showing the capsule with the afferent and the efferent arteriole coming in, and the Bowman, the proximal convoluted tubule leaving. And so this is known as the vascular pole, and this is the urinary pole. So here's a rare picture where those are actually found in the histology. Here's simple cuboidal epithelia, beginning the proximal convoluted tubule. Here's simple squamous. Now, to know that that was actually afferent arteriole, you would have to look at it at a higher power and see the renin granules. So I'm not going to require that you know that from the histology level. You can see renin granules in the light microscope.
Just a quick question. Do you have these online? So
I'm going to double check. I think I do, but I'll double check and I'll put them on this afternoon if we don't. Yeah. All right. So here, you weren't here for lecture, but here would be the pores. So we know that this is the plasma region, because these are the fenestra pores of the simple squamous, and these are the pedicels with the filtration slits. So this makes this the filtration region. Okay, so that's the level with an electron micrograph. And I might say, identify the fluid that you find here. Well, if it's the capillary, the fluid is plasma. If we're up here, it's still filtrate, because it hasn't become urine yet. Okay, so it's blood, plasma or blood in the capillary, filtrate in the capsular space. This is just a high-powered view of the proximal convoluted tubule, and you can see a little more closely the microvilli. So cortex, we know it's cortex, bless you, because over here is our capsule, and these would be the proximal convoluted tubules. All right, so for the distal, this would be distal right here, with a cleaner edge, okay? Usually not quite as large a diameter. So your histology quiz on Thursday could be from this slide, identify the region of the kidney. Is this cortex or medulla? Or it might be identify this component of the nephron, okay, which would be proximal convoluted tubule. These are probably all the same proximal convoluted tubule because it's so highly twisted. One slice through it is going to give us various transections, various views.
you said that distal is a cleaner one.
Yeah. So Here are some distals too, simple cuboidal epithelia, just a little bit of fuzziness, all right? Whereas the proximal is a little messier. This one is not quite as messy, but you can certainly see the difference in the length of the, obviously I can't use this slide because it has everything labeled. Unless I cut and paste and cover over. All right, now this one is from the medulla. Notice the absence of the capsules, all right? And this is a little easier to see, no proximal convoluted tubules either. These larger ones, and over here the cells were a little deeper, and so the cells are columnar than they are in the and closer to the cortex. So these are the collecting ducts. We'll talk about their role on Thursday and Tuesday next week. But they have simple cuboidal to simple columnar epithelia. And then we have the thick sections of the nephron loop, so that would be what these structures are here. And then we have the thin section of the nephron loop. Now, it's a little difficult to tell the thin section of the nephron loop versus the vasa recta, except that the vasa recta have blood cells in them. Okay? So these would be vasa recta, and these right here would be the thin sections. You can't tell if they're ascending or descending. Okay, so just nephron loop. Over here, again, collecting duct. It's hard to see ones with blood cells in them, but right along here are some. Okay, and the rest of these smaller ones would just be the nephron loops. And then finally, the epithelium that lines the minor calyces, major calyces, pelvis, ureter, and that's the transitional epithelium. Okay. So I wouldn't have you identify from histology whether it's a minor calyx or a major calyx, or you wouldn't be able to tell. The ureter has several layers of muscle, but we're not going to worry about that, the whole cross section, just the epithelium that lines it is our urinary epithelium. Okay? All right, so that's the histology kind of. So on the microscope, practice finding the cortex and the medulla, and then identifying the structures. You might actually find the macula densa. Let me show you one from our lecture. I didn't really focus on it in lecture, but you could see one in lecture. So again, here's proximal convoluted tubule, the messy appearance. Here would be distal. Here's our capsule, and right here, it doesn't look very tall, but it is distinctive from the rest of the tubule. This would be the macula densa right there, where all the nuclei are bunched up close together. All right, so even though it's not columnar in this appearance, you can still see the macula densa aspect of it. Okay? All right,